Hello everyone, my name is Michal Antala, and today I will present you my work for 4 years during my PhD regarding the peatland vegetation physiology and the measurements with the leaf and canopy level chlorophyll fluorescence with the experiment under manipulated environmental conditions. This work was done under the supervision of Anshel Rastogi and Marcin Strożecki in Poznan, and it was done within the project that was financed by National Science Center of Poland. My whole work consists of 4 articles, and I will try to go today briefly through them. So on the first article, which is review, I will explain you something more about the peatlands and what's happening with them. Then with the second article, I will try to introduce you more into the sun-induced fluorescence and its relationship with cross-primary production, especially under some climate extremes. And then the last 2 articles are the experimental work on the canopy level, is the third article, and the leaf level, the fourth article. Now you'll see better what I'm pointing on. So, as this was the whole PhD work, there were some goals. There were 4 goals, which each of them was addressed in one of the publications. For the sake of time, I will not read through the goals, but I hope that you will believe me at the end of this presentation that I was meeting them. And I will just move on to start with the topic. So why do we actually care about the peatlands, and why did I spend 4 years working with them? Well, it's everything about the climate change, as many things nowadays. And here you can see the 2 maps which are from the Intergovernmental Panel for Climate Change from the report from 2021. And they are showing how the different parts of the globe are warming with 1.5 or 2 degrees of the warming globally. And you can notice that on the top in the high latitudes in the north, the warming is the fastest. Now I will show you another map. And this map is showing the distribution of the peatlands. And my talk is especially about the northern peatlands. So it's this part more in the boreal region, ignoring the peatlands which are in the tropics, because these peatlands are mostly under rainforest. So I will be speaking about those northern peatlands. And if you compare these 2 maps, you can see that actually peatlands are in the places where the climate change and the warming is hitting the strongest. So the peatlands are one of the most impacted ecosystems. Also, if you see where there is the highest concentration of the peatlands, it's in the very remote areas. So I was working with the remote sensing techniques, and that's especially because we wanted to find out how useful is the remote sensing for monitoring of the impact of the climate change on the peatland vegetation. Just a couple of pictures from the vegetation, because I will be today speaking about the box cranberries, which are here on the left side, about the peat mosses in the middle, and then graminoids. I will be using this term, so just for you to know how the plant looks like, to get familiar with them, these are a couple of images. And I will move on to the first publication, which I said I will try to explain you on the example of this publication, what's happening with the peatland vegetation and how the climate change is actually impacting it, and then how the changes in the peatland vegetation impact the climate. So the strongest, the best result of this review paper is this one figure, which basically says everything. So let me walk you through it. And we know that the climate is changing in a way that the temperature is increasing, which causes increasing evapotranspiration. So that means increased drying of the surface. And there are changes in the precipitation patterns. Those are not conclusive, so in some places there is more precipitation, in some is less, but the patterns are changing. The climate changes are then impacting the vegetation. And in case of peatlands, it's mostly in favor of the vascular plants. So these were like the both cranberries from the slide before, or the graminoids. And there is less of the sphagnum. Also, there is a longer vegetation season for the vascular plants. This means that the productivity of them is increasing, and there is more of the easily decomposable litter. This litter is then used by the microbes, and the litter is then decomposed, and all of the carbon that was assimilated is actually released back to the atmosphere. These are not good news because the peatlands were storing for thousands of years the carbon in front of the peat, and it's being released. There are a couple of more the feedback loops which are shorter, which even make this case worse. But we found out already during the reviewing the literature that remote sensing is actually a good way for monitoring the phenology of peatlands, that it was already done before. What we can see from all of this is that the gross primary production is actually increasing. The problem is that the decomposition rate is also increasing. So actually the carbon assimilation itself, the net assimilation is then decreasing. And it's not just the carbon dioxide that is released from the peatlands, but peatlands are waterlogged ecosystems, so also methane is released, especially from the fens, which have higher water table. I will be also speaking about the chlorophyll fluorescence and showing you many results. For those of you who are not familiar with chlorophyll fluorescence, just a few sketches to explain what actually is the chlorophyll fluorescence. So when the sunlight is hitting the leaf, this light can take the three main fates, and that's being absorbed by the leaf or by the pigments in the leaf or other molecules. The light can be transmitted through or can be reflected. This reflected light is then also measured with the remote sensing and is used for either calculating different vegetation indices or then inverting the radiative transfer models. But this absorbed light is further partitioned between the leaf and between the photosystems inside of the leaf. And the light can be used for photosynthesis, but majority of this energy coming is actually dissipated as heat during the day, and some portion is emitted back as chlorophyll fluorescence. And here is just an example of the spectrum from the spectrometer. It's this peak here, which is showing this difference between the smooth line and this peak. That's actually the sun-induced fluorescence that I will show you in a couple of slides. Now, why did I start with this chlorophyll fluorescence? So maybe coming back, this light, the energy that is coming to photosynthesis and chlorophyll fluorescence is linked together in the photosystem. And normally we make use of the relationship between the chlorophyll fluorescence and photosynthesis to estimate the photosynthesis from the chlorophyll fluorescence. But if plants are under the stress, this relationship can be somehow broken, can cease to exist. And that was the case of quite well-known publication. So this was the publication by Martini et al. And my second publication in my thesis is a correspondence reacting to the findings in this paper. And I will try to now show you quickly what were the main points of this correspondence article. So the argumentation in the original article was that it's only the heat wave that is breaking the relationship between the fluorescence and gross primary production. But when this data was further examined, and that's especially visible on this figure, which there is a day-by-day time series of the fluorescence in red and gross primary production in black. So if you look closer on this relationship, even before the harsh heat wave hit the plants, actually the relationship was already quite weak because of the different course of the fluorescence and GPP. So you can see on this day-by-day relationships that the relationship in the scale of gray, these are before heat wave. So if you look at the day-by-day relationship, it was already very weak even before the strong stress started, but plants were already stressed. So why this was happening, we argued in our correspondence article that under the stressing conditions, actually stomatal conductance play a big role. And what we see, this decrease in GPP from morning 9 till afternoon 4, these are the points, is because with the increasing temperature and increasing water pressure deficit, the stomata of plants are simply closing and there cannot be gas exchange. And after the heat wave, so after this black line, we can see that actually the fluorescence is even decreasing faster than gross primary productivity of the plants. So if we reach stressing or strongly stressing conditions, we need to take this into account that the shift might not be the best in the linear correlations with GPP to estimate the productivity of the plant. Now I will move to the main part of my work, which was the experimental work in the site in Western Poland. In this site, there are two experimental sites, which we are calling CR and CL. These abbreviations are based on the dominant graminoids on the sites, and each site consists of 9 plots. In triplicates, these are controls marked by C, warmings marked by W, and warming and reduced precipitation marked by WP. The control plots are simply inserted frame into the peatland. The warming plots are with two kinds of warming. So during the day, it's passively by the open top chambers, which are basically glass houses or the greenhouses without the top. And during the night, there is the infrared heater that warms up the surface. So it's the all day continuously, so day and night continuously warming. The warming and reduced precipitation, the warming is done exactly the same way like before, and precipitation reduction is reached by those curtains, which are closing above the plot during the nighttime when there is precipitation event. The precipitation is cut only during the nighttime to not manipulate the light conditions. My third publication, so the first experimental publication, was done on the canopy level, and was using the field spectrometer, the Piccolo-Doppler system, which has a dual field of view visible on this picture. And inside of the cooling box, there are two spectrometers. So one of them is flame TV SNIR, which is measuring spectrum between 400 and 1000 nanometers with the cursor spectral resolution. This spectrometer is used for calculation of vegetation indices. And then there is a QE Pro FL, which is a very high resolution spectrometer and is used for calculation of sun-induced fluorescence. For our study, we used the SpecFit algorithm, which allows us to retrieve the full spectrum sun-induced fluorescence. So in this table, there are described the parameters that we can calculate. You can see in red, this is the full spectrum as retrieved by SpecFit. Normally, people mostly retrieve this O2A. So that was, if you remember a few slides back, this peak in the reflectance. So that's at this position of 760 nanometers. Then we have the O2B band, which is in the red region. And Then there are two peaks, so red and far peak.
Yeah, but I will check the مرة لكن like
Okay, that's probably not for me. Yeah, so we can also calculate the values at the peaks, and we can calculate also the full spectrum integrated value. To compare it with the gross primary production, we used measurements by transparent and opaque chambers, with measuring net ecosystem exchange with the transparent chamber, ecosystem respiration with the opaque chamber, and then we simply sum them to get the GPP as positive values. And there was done also modeling for the whole day ecosystem exchange or the gross primary production. What we found out with the measurements during the whole season, so these are the changes in the full spectrum of SIF for the two sides in the rows. So you can see CL and CR in different dates covering basically a whole year. So even outside of the vegetation season, starting from February, finishing in December. There are few findings that are visible on this figure. First of them is for peatland vegetation, the red peak is always smaller than the far red peak. This is different from the agricultural fields, where at the beginning of season was found that the red peak of the fluorescence is higher. For the peatland vegetation, which has evergreen plants, the case is that the far red peak is always higher than the red peak. Considering the impact of our manipulation, so the control is always red, warming is green, and then warming and reduced precipitation is blue. If you look at the CL side, you can see that for most of the season, there are not significant changes in the spectrum, but the warming and reduced precipitation especially increased the fluorescence during the June. So there was this faster increase because the plants started to be active sooner. That's how we interpreted this increase. But if you look at the CR vegetation, you can see that for most of the season, there is actually difference in the manipulated plots. Were showing stronger fluorescence than the control plot. This is partly phenological, but it's mostly because in the warming, there was faster or stronger growth of the vascular plants, and the vascular plants have then stronger photosynthesis and fluorescence. So what we see here is the change in the vegetation. This is just to show you statistically that these are the values of the integrated SIF. So the full spectrum is seen here, everything integrated and shown as one value to be able to check if there are significant differences. And as I was saying before, for the CR, we have for many of these points significant differences, while for CL, the differences are basically at the beginning of the vegetation season. If we want to compare how the SIF is actually doing in the estimation of gross primary production compared to the traditional reflectance-based vegetation indices, we compared them for the two sides. So the GPP is the gross primary production, LUE is the light use efficiency, and presented here is only R squared. But you can see that the O2A, so this is the far SIF, the traditional that mostly people are using, is actually performing very well. And it's outperforming even the integrated SIF. So we need to conclude that if we want to use simple linear regressions, actually the far SIF normally used is enough. Moreover, the vegetation indices like NDVI or NIRV have good correlations for the CL vegetation, which has more vascular plants and higher seasonality in the greenness changes. But for the CR vegetation, where the changes in the greenness during the season are not that pronounced, the vegetation indices are not performing well. The far SIF is still performing well. But what was new in this study, and it's highlighted in this red, was the multiple wavelength regression. And that's what I will explain on the next slide. So, because we have now the full SIF, which has many wavelengths, there are actually hundreds of wavelengths which can be used. And we don't need to only integrate over them. We can take the single wavelengths and we can check which of them and the combination of which of them is the best related to GPP. So we did this. We found out that if we combine those 10 wavelengths and feed them into the multiple linear regression, the relationship improves and the R square is improving, let's say, not that strongly because the relationship was already good with the far red only, but the RMSE is decreasing significantly. So the reliability of this prediction is actually increasing if we are using the information from the full SIF. And coming to the last publication, which was actually done on the leaf level, I need to explain a bit the methodology because this is different from the chlorophyll fluorescence, the sun-induced chlorophyll fluorescence measured on the canopy. So at the leaf level, we were using those darkening clips, which were put on the leaves or the capitulas of the moss, and were left there for at least 20 minutes to darken up the plants. And then the measurements were done with the fluor pen, which is from PSI, from Czech Republic, from the company. Two protocols were used. So in this case, it's not a single value of the fluorescence that we are interested in, but it's rather some dynamics of the fluorescence over time. In this OGIP measurement, this is the measurement with the high temporal resolution. This whole measurement is actually 1 second. So in milliseconds after switching on the light, the fluorescence is measured. And based on the changes, we can then calculate different parameters, which I will show you. And then the second protocol was MPQ protocol, which in this case, it's taking minutes, but it's measuring under the switched on light and then using the saturation process to be able to calculate parameters of the energy partitioning at the photosystem. So this is how the OGIP curves, the fluorescence transient curves, looked like for different plants. So we had brock cranberries and peat mosses, only those two because the measurements were there in the spring. And these are the plants that are of evergreen nature. So actually in the spring, in March, April, and beginning of May, when these measurements were done, only those plants are present in the plots. Other plants are simply dried. Looking only at the curves, you can see already that warming had a strong impact on Bob Cranberry, but to somehow translate these signals into some meaningful values, we had to calculate different parameters. Now, I know this is a lot of information here, so I will just point on some most important results. In the rows, you can see different plants. So this is for cranberries, this is for peat mosses. In the columns, these are different dates. What is important here, we can see the impacts of the warming. So this black full line in the middle, this is always control, which is 1. And then the relative change to control by warming is marked by this dashed line. Each step on this spider plot is 0.5. So that means if something is above this line by 0.5, there's 50% increase in the value compared to the control. If something is going inside of the circles, there's 50% decrease in the value. So what was increasing was actually the performance index and was the photosystem efficiency. Especially at the beginning of the season. As we go for the box cranberry from the beginning of season until May, you can see that the differences are decreasing. This is clearly sign that the plants simply started to be photosynthetically active sooner in the season when there is the warming applied. What were those impacts is improved photochemistry efficiency with decreased heat dissipation. So the non-productive dissipation of the energy was decreased. And this parameter, the absorbance per reaction center, is also important. We can see that it was decreased in the warming plants. That means that the reaction centers were just active. More of the reaction centers were active for the warming plants. If you look at the peat mosses, we can see that there is less of the differences. For the sake of time, we will just ignore this last measurement because the interpretation here is more difficult in this case because of the drying of the mosses. So those plants are more difficult to work with, I can tell it. But what we can see here is not that consistent pattern as for bark cranberries. There is not this disappearance of the differences as the time progresses, but it's rather going in some waves. This is just because the peat moss is more reacting to the temperature in the short time. And we reached the last figure. So in this figure, I just want to quickly show you how the energy pathways for the photosystem 2 in the plant are impacted by the warming and by different species. So you can see that actually mostly by warming, it's not significantly impacted. Only at the very beginning of the season, we can see increased quantum yield of the light intensity dependent energy dissipation and decreased the light intensity independent dissipation. This is again just showing us that the plants were activated faster when it's warmer. Quite logical result. But important in those figures are differences between the two species. As the regulated heat dissipation is much higher from coming from winter to spring in the mosses, and it's lower in the bog cranberries. So this shows us that the bog cranberries are relying on the non-regulated heat dissipation during the winter, similar to the evergreen needle trees, and they need some time of the warming, especially visible for this final. They need some time of the warming to get activated and to increase their photosynthesis. Whereas for the peat mosses, they have always low this non-regulated part of the dissipation and always high the regulated one, which means they cannot always just quickly react on any period of warming, even during the winter, and they simply switch on their photosynthesis. So coming to conclusions, as I was showing, when the climate is changing, when the warming is when the climate is warming, the peatlands are actually accumulating, or they have stronger photosynthesis, but they are accumulating less carbon because of the stronger decomposition. In short term, they are becoming lower sinks of the carbon. In long term, they may become even the source of the carbon. Vegetation indices are correlated well with the gross primary production in the peatlands when there is stronger change of the biomass over the year. However, if the peatland is dominated by lower vegetation and evergreen vegetation, we need to use sun-induced fluorescence. Far-red sun-induced fluorescence is good enough for the simple linear regressions, but better results are reached if we are using the multiple wavelength regression from the full spectrum of the SIF. When the weather anomalies become more extreme, the sun-induced fluorescence alone might not be enough, and we might need to use some thermal remote sensing to also include information about the gas exchange from the point of view of the stomatal conductance. And the the increasing in temperature is changing the seasonal amplitude of the sieve, as you could see on the example of CR vegetation for the full sieve. This is due to higher abundance of the vascular plant. All of the changes which we could see on the canopy level can be explained also by the physiology measured at the leaf level by the active chlorophyll fluorescence. Here are a couple of references, and thank you for your attention.